correctly, you are meant to turn those in on the day of the exam on Tuesday. So you're going to submit all of them, ideally all in one long document, but if you have to do 4 separate documents, that's fine. Well, it doesn't really matter. I guess I can just open it up either way. And I just make sure, I don't grade what you're saying per se, but I make sure that you have what looks like fairly complete answers to all of the lecture questions. And I'm hoping that's what you've been building through these in-person meetings, are the answers to that. So that work will be done, and also will help you study for the midterm on Tuesday. So if you could turn them in, and I think I still put 5 p.m. on my assignment, but I didn't write it here. That same day that you take the exam. So you do get points for turning them all in, as you see in the course description. And that's going to be the pattern for the semester, that you'll be turning those in as a block at the day of the exam. The other thing that will happen on that day is I will be posting, or possibly just sending you emails, or both, which essay you need to turn in that day. So if you're a procrastinator, you're going to have a lot of work cut out for you, because you'll be taking the exam at 9:20. You'll have an hour and 35 minutes to do the exam. I think I set it up. I just set it up, but I set it up, I believe, so that the time the window is open for Confer Zoom is even longer. So I will be in Confer Zoom, and you can pop over to that window and ask me questions through the chat, not aloud, so you won't disturb other folks. I don't typically, my emails sometimes pop up quickly, but if you send a question through my email, I may miss it because I'm in the Confer Zoom window. If you have a problem having both windows open, just let me know before the exam starts. So I'll have Confer Zoom open 10 minutes before, so at 9:10, so you can tell me whatever your particular needs are, or if you're having some sort of technical problem. We have about 10 minutes to try and work on that together before the exam becomes available. So when you go to take the exam, you'll go into quizzes, and at that point, you'll see the exam should open, and it'll be open for an hour and 35 minutes. You can, you have the ability to go back and forth between questions. So what you'll see is that there are a number of short answers. There are actually 10-point short answers. There's some multiple choice, and I think that's the only 2 formats that you'll be dealing with. So please time manage yourself. You might want to take a look at the whole thing first. I'll probably, in fact, post in the description how many of each type of question, so you'll have an idea for your time management. Because that's the hard part. One question shows up at a time, and if you get so entrenched in one 10-point question that you're giving me a 20-point essay, then you may run out of time for the remaining part of the test. And then you know yourself best whether you want to do your multiple choice first or you want to do the short answers first. That's up to you. So you can go back and forth there. I did put in the policies that this is a totally closed book test and closed screen, other than the one you're taking it on and Confer Zoom. So if I see any evidence that things have been cut and pasted, and I did have that experience last spring, that things were cut and pasted, then I will give you a 0 on the entire test, not just that particular question. So one thing that it is a kind of red flag, and I encourage you guys to work together in writing these essays. You can, shift them back and forth between each other, or the short answers, the lecture questions. But please try to come up with your own wording, because that tends to be a red flag for me if I see the exact same terminology, the exact same verbiage for the same question. Now, this comes up even more so when you're turning in the essay that day. So when you find out which of the 7 essays it is and you turn it in, first of all, I'm not concerned with format. I would like it typed. If you're doing some sort of flow chart, or you have to draw a graph and you embed that in, it's hand drawn. That's fine if you don't have the computer skills to do something as complicated as that. But that should be supplemented by typed. You have to realize that I'm going to be reading so many of these. I do have a rubric for them, so that I am looking for all parts of the question to be answered, and to be answered fairly fully. So that's the formatting for next Tuesday. Are there any questions? You can either put them in chat, I've got that open, or you can just unmute yourself and ask about that.
Sorry, Professor, I missed the first minute or two. So you said we have to keep the Confer Zoom open as well when we're taking the exam?
I'm suggesting that. You don't have to do it, but it is the way in which you can ask a question most efficiently, because I'll have that window open, I'll have the chat open. I'm encouraging you to use chat to ask the question rather than, unmute yourself so you don't disturb others. But as far as my email window, it just pops a little message up really quickly while I'm in Confer Zoom. So I may miss that. If you get desperate and you can't use Confer Zoom and Canvas at the same time, then just let me know that before the test. I do have another computer, but my eyes will probably be on the Confer Zoom screen, so not the email. But I'll try and periodically check that if you tell me that's the only way you can do this, that maybe you only have one screen to work with. Is that clear, Prabdeep? Sorry. Okay. All right, any other questions? I'm getting a few in chat. They will be organized by type, so they'll all be together. And you can go back and forth between questions. Dr. Sundar and I went back and forth on that policy because I think it's the best way to take a test, is to be able to go back and forth, so that especially when you're trying to time manage, but it makes it where there is more of a temptation to cheat. So I encourage you to utilize it in the proper way, because I did catch people last semester. They had either copied or pasted from the internet, and things that are submitted through Canvas, there is the turn it in feature within Canvas. So it does search for stuff like that. And then when people were using exactly the same verbiage as their study partner, I also was kind of red flagging that, or directly from the book. I got some incidents of that too. So just put it in your own words. That's all you need to do. Okay, any other questions? Okay. All right, so we're going to finish up these lecture questions, and just in case other people came in a little bit late and missed the beginning, I want to remind you, you're turning those in on Tuesday, and I believe I said by 5 o'clock. I think all the assignments other than the tests are 5 o'clock. So you're turning in complete answers for 4 sets. That is from the beginning of the course through the ones we're finishing up today. So I'm hoping you've built that. And then you are also turning in an essay. So I have noticed a number of people seem to be struggling to turn in even the couple slide drawings that are due by 5 p.m. I'm getting them at 10 o'clock at night that day. See, I've only been taking a few late points off, and that's going to hold true for the lecture questions as well. I'll just take some late points off. If you're turning in the essay that's part of the sort of take-home test, though, then I may have to look at that a little more severely, since it's not really fair to the others, since that's that's worth much more points, point-wise, and it is really a test. So I highly recommend that over the weekend, over the next 4 days that you have written those answers and you have them in a typed form. If you're not, hopefully everybody can type. That's another thing you may want to give me a heads up if you can't type, because Canvas, you're typing the answers into Canvas for the quiz. For the essays, if you've got them handwritten, then I think it's a great exercise to handwrite them. And I think in the past, Dr. Sundar's even required them to be handwritten. I think it's a great exercise to retype those handwritten, to type up those handwritten, because you will be going over it and reviewing it, and you may decide to change some of the wordings or add some things you now know, now that you went further into the material. So the last thing I'm thinking of is I haven't posted feedback for the last 2 essays. I don't mean feedback, but my old feedback comments, the sort of outlines to help you. And I will do that by the end of today. So we'll have finished, we finished 6 already. 6 is very straightforward. It's big, but it's straightforward. 7 is a little bit been quite carved up, and there's a lot of editing from the textbook. So that's one you may need a little bit more feedback, a little more guidance as you go through, and that outline might help you more. Okay, so if there are no issues or questions, then what I'd like to do is return you to your breakout rooms. I believe there was one group, I made it a little larger than before, that was working on lecture question 12, 13, 14, and 15, and that's a lot. Partly question 12 and question 15, they overlap so much. One says, basically, not 12, 13 and 15. One says, what causes the initial platelet activation degranulation, and the other one, what increases it. So those two are very, very similar. If you'd like in your group, you could break it up into what seems logical to me as maybe one person discussing the evolution of platelets in the bone marrow, and maybe another person discussing 13 and 15, and then finally one third person possibly discussing what's in these granules that the platelets are going to release when they're degranulated, because that's kind of a big topic. It brings up the phases of blood clotting. That what I mean by that is the initial vascular spasm, and then platelet aggregation, and then finally coagulation, because platelets aid in all of those steps. And then we'll go into coagulation. So I don't think I broke anybody up into a group F last time, F and G, because I kind of knew that we won't get all the way through hemostasis. So for group F, I'll call them group F, since the last time the other group was E. You're going to be looking at 16 and 17. And so you'll be looking at the intrinsic and extrinsic pathways that cause coagulation. And this is a little sloppy in their terminology, because it'd be nice if we could really just say intrinsic is within that organ and extrinsic is sympathetic or endocrine. But here what they're saying is intrinsic is within the vessel and the blood itself, and extrinsic is anything outside that blood vessel, meaning the tissues that the vessel's running through. So even though I said it would never mean anything else, here it is meaning something kind of else. And then there's some, in those two pathways, there's some negative and positive feedback loops. And the book is pretty good at pointing those out. So you should have been able to find those. And then we'll go into the last breakout room, G. And here now we're looking at, okay, now the body is healing and this clot needs to be removed. So that's the last of the lecture questions by fibrolysis. That's the breaking down of the clot. And then it's sort of as a separate question, but not unrelated. What are the roles of the endothelia cells in preventing inappropriate clot formation when you don't have damage to the vessel? And then what I'll do is probably supplement that, because I've made a figure that's shows all of the various roles that endothelia cells have played. Those are the tunica intima cells that line both capillaries and arterioles. And there's quite a list to that. And we're going to even add more to that when we come back after this exam. So, so let's break you guys up, unless you have a, I'm not sure, since I had it randomly break up I'm not sure I'm going to end up putting, maybe we'll just take a few minutes. And if you can let me know if you were in group E, I will make group G and F, and then I'll check my chat and I'll put whoever isn't in, actually I can't do that, can I? Why don't you just let me know if you were in E? Okay, I see people popping up. Anybody else? I've got 4. I know there's more than that. If you haven't used chat, it should be down at the bottom of your screen. Change this to manual. Okay, getting them a couple more. All right. How about if I give you another 2 minutes to tell me, and then otherwise you're just going to randomly be put in that group, because I only have 5 people right now. Maybe that's all that was in there, but I thought it was kind of 6 people per group on Tuesday, and then I added a couple extras to group E. All right, let me rename it while I'm waiting those couple minutes. All right, so let me assign to group E the people who have told me they were in it. Let me see if I can move this over. All right. Yes. One more. Oh, and Kelsick. Okay. All right. Then I'm just going to sort of randomly add some people to group E. So how many have I got in there? 2, 4, 6, 8, 11. Maybe I'll put one more. This is a big question. Okay. Now I'm going to just do the rest of you. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, So, here's the last one. All right, here you guys go. You have 15 minutes and just pop out.
Recording the lessons like before she starts them or not?
Okay. All right, so now it looks like most of you are back, and or maybe all of you. So I wanted to start with Group E. Do you have a spokesperson for the lecture question 12?
Professor, I'll speak up for 12, 13, and 12 and 13, and 15 will be answered by someone else.
Okay.
Okay. So for 12, the answer is thrombopoietin, IL-6, and CSF, which is a colony-stimulating factor. They stimulate myeloid to form the megakaryoblast, which is an undifferentiated stem cell. So this matures into a megakaryocyte, which is a cell, and then pluck off into pieces to form the platelets.
Okay, that's good. There's a couple things that, I wanted to add to that, partly because I think it's kind of cool, but let me share a screen with you. So thrombopoietin, which notice that root of the word again is poietin, which of course seems to be linked to any terminology that's important for blood clotting or for blood development. And so I made a little figure here. Thrombopoietin is made at a constant rate. So, we're so used to a negative, this is the part that I just think is interesting, negative feedback loops that what happens is that you have some receptor and then a control center, and the control center then releases a hormonal signal or a neural signal that affects the target. Well, they don't go through this negative feedback loop in the book, but in fact, platelets are sort of their own negative feedback loop. So I actually don't know if I know the source of the thrombopoietin. It's probably the bone marrow, but I'm not sure. But it is released in a constant fashion. So the signal that tells the bone marrow to make megakaryocytes, like Kao Sang said, is released chronically. Platelet number is then generated from these megakaryocytes, which we don't really have in the lecture question, so maybe I'll point out a little bit more about that. But these platelets then have on the outside of them, and I did this kind of crudely with little black lines here. Just go back to black. These things are an enzyme. So it's a membrane-bound enzyme, and the enzyme's job is to destroy thrombopoietin. So when you have sufficient numbers of platelets, the signal to the bone marrow to make more platelets is diminished because the platelets are constantly destroying the hormonal signal that would cause them to make a megakaryocyte as opposed to making a erythroblast or one of the white blood cells from that lineage. However, when platelet number drops, say after you have a blood clot, and those platelets, once they become activated and differentiate and degranulate, they are gone. They're destroyed. Then you have low platelet numbers, and that means that there aren't enough enzymes to destroy the thrombopoietin, and thrombopoietin levels rise, and then the hemocytoblast and the myeloid stem cell are getting signals it's time to make more megakaryocytes. So I thought that was kind of cool. Now, the other signal, as Kelsang said, are these colony stimulating factors. So those are just a general big category for lots of different signals. So under that, we already know of EPO. That's a colony stimulating factor, clearly that will eventually lead to becoming red blood cells. These colony stimulating factors are talking to the hemopoietic stem cell and the hemocytoblast, whichever you want to call it, and it's talking to the myeloid versus lymphoid cell lines that we're going to come back to after this exam and look at again. And then we just saw that we had thrombopoietin as another colony stimulating factor that leads to platelets. But it mentions, and Kelsang mentioned, that you have another colony stimulating factor, interleukin. So when you see IL means interleukin. And we're going to hear a lot more about these guys after we finish this test and when we enter into the immune system. So there are multiple sources of interleukins. The ones that we're going to be talking and focusing mostly about are during an immune response. So you can imagine that during an immune response, you might need more platelets. So this is from an immune stimulus, and it's going to then lead to more platelets. I think it does other things besides more platelets, if I recall well, but at this point, we just have to know it as a colony stimulating factor. Oops, I forgot the 6. Then you'll say, why do you have a 6 after that? Because there's greater than 35 different interleukins. This is a category of substances, different interleukins. So what about these megakaryocytes? I go over this in the lecture, so you should be aware of it, even though it's not in this. There's no essay on it, and there's no lecture question that's directly asking that. But remember, these are these gigantic cells. That's how they get their name, megakaryo. They're a large cell body. And what they're doing is, if this is the endothelia of the bone marrow and the blood is going through here, these guys sit on the tissue side of the bone marrow, and they project little extensions of themselves through the paracellular gap between the endothelia, and these little extensions then break off. So you do need to replenish these megakaryocytes because they are going to be eventually, releasing into the blood most of their cytoplasm and plasma membrane with the granules that they've made. So they make a wide variety of granules. Our book doesn't cover everything that is contained in these granules, but that's what these platelets are. They're little packages of granules that the megakaryocyte has generated and filled this cell with. Now, what is a granule? A granule is a vesicle, so it's a little membrane-bound packet of lots of contents. And I think the next answer, which it sounds like the same person is going to do, has to do with activation and degranulation, so that when the platelet gets stimulated to start a blood clot, then it's going to release the contents by exocytosis into the plasma or into the interstitial fluid that the platelet finds itself in. Okay, so let's go on to 13, which has to do with the various stimuli that the platelets can listen to, or conditions that the platelets could be under that might lead to them being activated and start to release these granules. And is that you again, Kelsang, or is it someone else?
Yes, Professor.
Okay.
So the question 13 answer is when the platelets contact the collagen fibers, which is normally hidden by the endothelial cells. Platelets are activated, and platelets become activated when they contact any thrombogenic surface, such as the injured endothelium, subendothelium, and artificial surfaces, and or they are exposed to soluble activators. The VWF made by a normal endothelial acts as a bridge to link other platelets to the initially activated platelets, and this lead to granules exocytosis. The third answer for the contents of bioactive substances are the serotonin, thromboxane A, ADP, calcium, hydrolytic enzymes, PDGF, the growth factor, and PF3.
Okay. So in your list, you had serotonin, thromboxane. What else?
One second. Thromboxane A, ADP, calcium, ADP.
And the growth factor, right?
Yes, the growth factor, hydrolytic enzymes, and PF3. Hydrolytic enzymes.
Okay, and I'm going to put the hydrolytic enzymes down here.
Okay, and PF3.
I'm going to put PF3 up here. Put it in parentheses. Enzymes. Okay, so why I put these in different lists is because the source of all of these things, platelets, right? Other platelets.
Yes, Professor.
Right. So it's kind of an answer we've already approached, but we can revisit it. The answer to 15, which is once you've activated a platelet. But as Kelsang said, the initial activation has to do with the binding to collagen. There is a factor called von Willebrand factor, if I'm saying it properly, that helps this along. So this stands for a V, sorry, it doesn't look much like a V. Von Willebrand factors, named after obviously a person, but because there are genetic conditions where people are deficient in von Willebrand factor, and then they have trouble, but it's not impossible to cause blood to clot. Now, why they can is in part because there's more than one type of Willebrand factor, so that genetically you're not deficient in all of it. And that was, of course, not known when they named the disease. They had them all under the same umbrella, but now we know there's actually more than one type of Willebrand factor and more than one source of it. But the main reason they can still, they're not as efficient, they can still cause blood clotting is because you have other stimuli. So, for example, these hydrolytic enzymes are going to come from damaged cells. And those damaged cells could happen due to an injury. Collagen itself, without the von Willebrand factor, can bind a platelet and activate it. It just doesn't do it as efficiently. So why don't we spontaneously throw blood clots? Because there is a number 4 here, and that is that slowing of the blood flow or pooling of the blood where it's not moving will cause platelets to activate as well. They don't even actually need something outside themselves if they go slow enough. if they become in contact with each other, so you could think of them as a whole bunch of cars and traffic, and as long as they're speeding along and they're not in close contact with other cars, they are actually looking at suppressing agents that are being released by the endothelia that are healthy. They're kind of being exposed chronically to red lights that say don't release your granules, because the platelets are really in a hair trigger kind of fashion. They take very little to start to activate and degranulate. And once they do, then just like Kelsang said, these other platelets are going to cause more platelets to activate. So what sort of situations could that happen? Well, here is one situation where you actually have damage to the endothelia. And that would be, say, if you cut yourself, you could have damage to the endothelia, and that would expose the collagen. And if it's a deep enough, you really cut this capillary bed, of course, there's all the extracellular matrix with its collagen around the tissues as well. So platelets that leak out of the bloodstream and into that tissue space are going to encounter collagen. In the blood plasma itself, there is von Willebrand factor. That von Willebrand factor is secreted by the endothelial cells. The endothelial cells also hold granules full of von Willebrand factor, and when they become damaged or triggered, they can release more of it. So the interesting to me thing, I kind of researched this while you guys were doing your breakout rooms. What the textbook says is von Willebrand factor is in the plasma, and it doesn't give the endothelia cell any role in it. But 80% of the von Willebrand factor is made by endothelial cells. So endothelial cells generate this von Willebrand factor, secrete some of it, and keep some of it in granules. Meanwhile, again, the book doesn't talk about this, so I wouldn't test you on it. There are enzymes that break down von Willebrand factor in the blood. So you can see, kind of like with the platelet generation and thrombopoietin, there is this delicate balance where you have enzymes destroying the von Willebrand factor. At the same time, you have it being released by endothelial cells when they're healthy. Now, it only becomes really active when it sees collagen, and then it forms this bridge. So it'll bind the collagen, will bind platelets, and increase platelet activation. So what about the other part of the von Willebrand factor? Not all of it comes from endothelia. Well, it also, and I have to wake my thing up again here, it also comes from platelets themselves. Oops, sorry, it's acting a little slow. I'm going to erase all that. I don't know why. I guess when I woke it up. Let me go back. Von Willebrand factor. And the other interesting to me part of this is that the von Willebrand factor, and we'll say it's a different type by putting an asterisk next to it, that is released by the platelets is not as susceptible. It's not that it can't be broken down by the enzyme, but it's more resistant to the enzyme. So why I'm going through this is not so much so you have that in your essay, you're welcome to include it, but that platelets themselves promote greater platelet activation, and endothelial cells and the conditions of the plasma when you have intact blood vessels are constantly suppressing everything about platelet activation. And you have this delicate balance that your blood is fighting all the time, because if you have a spontaneous clot form, that's called a thrombosis that we'll talk about later, due to something like the slow flow or pooling of blood, then then that can be a life-threatening condition. But on the other hand, you really want platelets to activate without a lot of stimuli and have a lot of redundant stimuli to activate them because they're so critical. In fact, you really won't do good coagulation without activating platelets. So what sort of situation would cause blood to flow slowly or pool? Well, surgeries do that because when the surgeon goes in, they're going to cut veins. They're going to try not to cut arteries because, of course, arteries are high pressure, but they're invariably going to cut veins or venules to get at what they're trying to get at. And that means that when the person's healing, the venous return from that area is going to be impaired, and blood is going to go through the capillaries, not find a way through that set of venules. They'll probably, there's a lot of redundancy, find eventually a way through another set of venules, but blood will slow. It won't be exposed to as many of the suppressing factors that the endothelial cell are releasing, and you could potentially get a platelet to activate, which will then cause a increased activation of other platelets. So that's the portrayal of how you activate platelets. So question 14 is going into, we've already listed quite a few of them, but not all of them, and not all of the roles platelets are going to be playing in blood, the three phases of blood clotting. So who's going to be doing question 14 about what else is in platelets?
Professor, that would be me.
And that was Manreet?
Yeah.
Okay. So maybe I'll leave this one up and then we'll just, I'll just keep adding to the list as you go through it. Or I don't know, why don't you start and then we'll just see how this goes. I can go back and forth.
Okay. But actually, a lot of the ones that we talked about are like on there. Like, I don't know if we have any more that haven't been mentioned already. I don't know.
But the roles are more.
Oh,
right?
okay.
Yeah. So that's why I was thinking maybe I should do it separately.
Okay.
So why don't you just go through them again? Okay.
Okay. So we have first serotonin and thromboxane A, both of which cause contraction in the vascular spasm phase. So that will create a positive feedback loop because an increase in serotonin and thromboxane A will increase prolongation. We also have ADP, which is also a positive feedback loop because it will cause other platelets to become activated and they will connect platelets together to physically make the platelet plug. We also have calcium, which aids in contraction. That is also a positive feedback loop since, as we learned before, the more calcium you have, the longer the contraction lasts. Calcium helps in all stages. We have hydrolytic enzymes, which are a clotting factor. And we also have growth factors, which are important in the healing process, and PF3, which is on the platelet membrane and is critical for coagulation as it has negative charges that act on the site for clotting factors to become activated enzymatically.
Okay, that was very thorough, and I apologize. This thing is acting so slowly. I think I may just try turning off my share for a moment and see if when I share again, it gets better. But she did a great job. And you'll notice that in my lecture notes, I was a little oversimplified. Manreet covered it well in the fact that many of these factors that are released have multiple roles. So for example, Kalsang mentioned that thromboxane A helps in the aggregation of platelets, but it also helps in the vascular spasms. So just, make sure that you understand, even though it's not in these lecture questions, it is very directly in the essay that often what happens when you cut yourself, say, is that you're going to cut some small arterioles as well as capillaries and, the surrounding tissues. So you're going to initiate a vascular spasm. And that's going to be often an injury response at first from the muscle tissue, the smooth muscle of the arterioles and the venules for that matter. And they will cause contraction, but that contraction won't last very long. It needs a positive feedback, as Manreet said, in order to cause it to be prolonged. So why do you want to prolong it? Why would you want to prolong your vascular spasm? What would be the reason for doing that? Any ideas? Anybody could just either type
You
it
want to
in.
like basically have enough time to to get more platelets to get added and form the plug.
That's right, because if you don't, you're going to be losing blood, right?
Yes,
So
it's a
you're not only going to be losing blood, but you're going to be potentially having pathogens enter into this opening. So one of the goals you'll see when we get to the immune system is, and we talked about with skin, is you want to keep your blood vessels far away from any potential either normal resident bacteria and viruses and fungi and such, and you don't want to have any opening into that because your bloodstream's a highway system that pathogen can reach the rest of your body. So by closing down blood flow through that particular area that's been injured, you're going to prevent you from losing blood, but you're also going to prevent or help prevent pathogens from entering into your bloodstream. So you prolong it, and I think the book says it can go like 20 minutes with these positive feedback loop from calcium, which of course helps contraction, and thromboxane A and serotonin. Some of those same things, as Cal Sang mentioned, also help increasing platelet aggregation. And as Sana just said, why you want that is you want to make a plug. You want to put your finger in the dike by making at least a temporary plug at the site of the damage. So during this platelet aggregation, lots of other stuff is released that's going to be used later. And one of the ones that Manreet mentioned that we're going to really focus on is going to be PF3. And I still prefer calling it PF3 because I think that the the full name of it is confusing, easy to confuse with other terms. So in my figures, I kept calling this PF3. So we're going to visit that again when we do coagulation. The other thing we'll visit again when we do coagulation is calcium. Calcium is going to play a role in all three of these stages, the vascular spasm, platelet aggregation, and it's going to be critical for coagulation as well. Let's see, what else? I think we've touched on most of the things that were on our list. The growth factor, I don't mention a lot more, the PDGF, platelet-derived growth factor, that stands for. Growth factors in general will help with healing by, making cells more metabolically active so that they can synthesize proteins and such. And it does seem to have encourage platelet aggregation. So nowhere in this, maybe the next group who talks about 15 can really describe what happens to platelets when they aggregate. So who will be doing question 15?
I will. Jessica?
Oh, great. Thank you, Jessica.
Actually, for our group, when we answered question 15, we had a lot of overlap with question 14.
Mm-hmm.
So for 15, it says what factors act to increase platelet aggregation and activation. So we put down serotonin and thromboxane A, ADP, calcium, growth factor, PF3, and we also included VWF.
The WF meaning VW.
Yeah.
Yeah.
which Kelsing mentioned before. It acts as a bridge to link other platelets to the initially activated platelet. And then also prothrombin activator converts prothrombin to thrombin, which acts in a positive feedback loop to increase platelet aggregation.
Okay, so we're going to have a bunch of positive feedback loops happening. So that was a good addition you had there, Jessica. So thrombin is something we haven't talked, oops, it was up here again, I guess. Thromboxane, oh, sorry, this is supposed to be thromboxane. That is thromboxane A. So thrombin is not made during the early part of platelet plug formation. It's made after you start coagulation. But it reinforces the platelet plug because, as you'll see when we get to coagulation, there's quite a lot of reactions that have to occur, and all those reactions take time. And when during all that time, you're trying to prevent any entry of pathogens and prevent loss of blood. So platelets, when they activate, start out as, these little chips of cells. But once they activate, they turn into long branching, maybe I'm exaggerating because they can't get bigger in volume, that reach out with extensions that then touch other platelets that are activated or the unactivated ones and cause them to activate in a kind of chain reaction formation. Now, one of the things they're using, as she mentioned, is this platelet factor 3 that's going to develop. This is a membrane-bound phospholipid. So it's actually not a protein, it's a phospholipid that's inserted into the activated platelets by their cells. And it's going to play a role in aggregation, to help aggregation occur, and it's also going aggregation. And it's also going to play a role in coagulation. I'm sorry, what did you say?
Do you mind sharing the
Oh gosh, here I am writing away. I finally got it working. Thank you, son. Here, I'm still getting used to this online teaching thing, Mom. Okay, here we go. Okay, that's better now, right?
Yes.
So I tried to take notes as she went. Serotonin, thromboxane A, PDF, that's the growth factor. Calcium, von Willebrand factor. In a way, you could think of this as a kind of positive feedback too, even though it wasn't on the list. But from what I just told you, platelets release the von Willebrand factor, and that tends to cause better binding to collagen and reinforce. So that's kind of a positive feedback. And then we have thrombin as another positive feedback here, where once you generate thrombin, and you need activated platelets to do that, you get greater platelet aggregation, more platelets get activated. So here I was trying to exaggerate my drawing, but platelets starting as these little tiny chips of things, this is where I made it too much volume, but they grow these long extensions that reach out to touch other platelets. And when they do, they cause those platelets to activate, which touch other platelets that cause those platelets to activate. So once again, another big positive feedback loop. When they become activated is when they sprout this platelet factor 3, which is the phospholipid in their membrane. And it does help encourage aggregation, but it's going to be critical for coagulation to occur. So in some folks, they have deficiencies in platelets. And when you have deficiencies in platelets, as you'll see in the next topic, that means you really won't be able to very easily do coagulation at all. So you can obviously make a platelet plug because there's lots of overlapping and redundant ways to make a platelet plug, but platelet plugs are fairly unstable and they're not going to last as long as it takes for you to heal that area, because you know how long a scab is on you. It's not just a matter of hours, it's days or longer depending on the depth of the wound. So I think that's all of Group E. Did we do everything in Group E? Did anyone want to add anything from that group? Okay. So I'm going to stop sharing this. And I probably should have opened this up already, just so we have a picture for when no, that's B, yeah. Let's have a picture of the intrinsic and extrinsic, because this is where the real editing happens. So I thought I would open up the lecture slides and share that picture. Why are they not opening? There they are. Okay. Guess my poor computer, it's struggling to do everything I'm asking it to do simultaneously. Okay, so do we have a spokesperson for Group F?
For this question, that'd be me.
Okay, great, Sana. Let me screen share so we'll have sort of a visual while you're talking.
Sure.
Okay. All right, so here is, not now, here is hemostasis, and we're going into this third thing, which is coagulation, right? So this question asks you to compare what's similar and what's different between intrinsic and extrinsic pathways. So here they are.
So for similarities, we got that they both require the clotting factors PF3 as well as calcium. They both pathways lead to making the prothrombin activator, which changes prothrombin into thrombin, and then goes on to do the rest of the cycle. For differences, we got that the intrinsic pathway requires trauma inside the vessel, and in that case, local paracrine molecules are released. And for the extrinsic pathway, that is stimulated by trauma externally, and it requires interstitial tissues to release a paracrine signal, which is the tissue factor. For, and another difference is that intrinsic pathways are slower while, because they require so many more steps than the extrinsic pathways.
Great, that sounds fantastic, which you now know, this is also an essay, so you want to deal with that. You probably want to acknowledge that they each require clotting factors, but some of those clotting factors, without naming them, they share common clotting factors, but they also have different unique ones that only one pathway or the other are using. So I know I just lumped them together as a variety of clotting factors, but when we look at them more detail, you'll see that they share some and they also have unique ones. These clotting factors are largely made by your liver, so they're already in your plasma. They're like little, since I have a son, you probably relate to this. They're like little Lego pieces that you can combine with other Lego pieces so that eventually they form through a enzymatic cascade. They form, meaning one step leading to the other, they form eventually something that can called prothrombin activator. So the more details of that are clearly shown here. And I have edited out knowing any of these Roman numerals, because who's going to remember those, a week after the exam? They're going to be gone. And I believe that Sana mentioned this, but notice calcium, and there's even calcium needed later. Without elevated levels of calcium, it's difficult for these to occur. Now, the interstitial fluid, the plasma, it's carrying calcium, but it's not at very high levels. So the supplement of calcium from platelets is important. And you're seeing here platelet factor 3 being used on the intrinsic, but later you'll see it's also used in their common pathway that both require. So just like Sana mentioned, the whole reason that this is faster, or the main reason, is that tissue factor can skip a whole bunch of these early cascading reactions. It's just going to take 7 and make it into 7A. Well, that also happens in this pathway. That's what that arrow is meant to show you, that that's a common clotting factor. But you get this complex, whereas here you need to go through these further steps before you get something that can then enter and act on clotting factor 10. This is clearly the more complicated pathway, and there are people, as you probably know, with a condition called hemophilia. And philiacs are genetically deficient in clotting factors. There are different types of hemophilia. One of the more common is deficient in this number 10. So you can see that if it's deficient in this 10, that they're not making it, that would affect whether you're intrinsic or extrinsic using the pathway. So why have two? Well, why have two pathways? Well, I'm flinging my hands around here, waving them around, but my guess is it's one, so critical to be able to cause your blood to coagulate. And number two is that you do have different situations where coagulation may be useful. I mean, this is what my theories are, at least, that this pathway would be for the blood that is leaked out of the vessel. It's gotten into the interstitial space, whereas where Sana mentioned accurately, this tissue factor would be being released. And therefore, it's forming the clot on the outside of the vessel. Whereas this is forming the clot on the inside of the vessel, where you have more of the clotting factors, which are found in the plasma. Now, these aren't all found. I shouldn't list them all. This one becomes that one, but you know, the ones that you're starting as raw materials, they are found in the plasma. And then you have platelets that could be releasing platelet factor on both sides, but you know, more of the platelets are going to be inside the blood vessel. So because of this intrinsic pathway, you have the danger of thrombosis, though. You pay a price for having a clot form kind of on both sides of the damaged vessel, and therefore be a stronger clot. But that also means that under the right conditions, if you activate platelets, you do have this intrinsic pathway that can cause a clot to form inside the vessel when there isn't any damage to that vessel. Now, this is being suppressed, as we'll hear shortly, by the endothelial cells. So I put steps 1, 2, 3, but you know, it's up to you whether you you could break it down to more steps if you want. So then it enters in, when it makes this critical prothrombin activator, it enters into a common pathway, as Sana mentioned, this one, where prothrombin becomes thrombin. So remember, this feeds back positively, causing more activation of the more platelets to cause greater aggregation. But the other thing that happens is there's a positive feedback loop here as well that we haven't talked about. And the next person, is that you again, Sana, or is it somebody else that's doing 17?
I think that's me again.
Okay.
17. Yeah, so for the positive and negative feedback loops, for one positive, we have thrombin, and it's a positive feedback loop as it increases volume and also increases platelet aggregation as well as aggregation.
Whoops, we lost you.
And then there's also a negative feedback loop with the fibrin, and it has to do with thrombin as it binds up free thrombin, and that decreases the rates of the earlier two steps, which will allow for clotting not to happen all the time and also decrease platelet formation. So we had those two, but we weren't really sure what the last positive feedback loop would be.
Did I ask for 2 positive, right?
Yeah.
So we did cover 2 because this guy positively feeds back to cause greater aggregation. You also get this positive feedback in that it speeds up the production of prothrombin activator. So that's where I just made it as a single arrow here. But what happens is thrombin can go back and potentiate these reactions so that they happen faster. You get more prothrombin activator, which makes more thrombin. So thrombin's kind of that central role with both increasing aggregation and increasing the production of prothrombin activator. Good. So we'll get to the negative feedback loop when we get to the next slide. The other thing I'd like to point out is there's positive feedback in platelet aggregation. So you could come up with more than the 2 that's asked, right? This is asked for coagulation, but you get positive feedback when you do the vascular spasm. You get positive feedback when you do platelet aggregation, whereas one platelet that activates will cause others to activate, which would then cause aggregation. Platelet factor 3 is important in doing that, as well as the other things that are in the granules that we talked about previously. So you can see this is kind of a big snowballing effect. And you need to have, with all these positive feedback loops, there has to be a break on this. And we're going to be talking in the last group about the endothelial role as a break, but the healthy endothelia. But we also will have thrombin, as Sana mentioned, as a break. Because as coagulation begins and you form thrombin, you could think of thrombin like a big sponge, and fibrin as a big sponge. And as fibrin is formed by thrombin, this thrombin is acting as an enzyme, to take fibrinogen to make it into fibrin. And as this enzyme's acting, it forms a fibrin, and that fibrin, which are protein fibers, but they're insoluble. So you can imagine this thing growing as insoluble fibers, like collagen is an insoluble fiber, big fibers that are forming both inside the broken blood vessel and outside the broken blood vessel. These fibrin start to bind up thrombin. So we always think of an enzyme active site, quickly binding, doing its job, unbinding, going away. And I don't know that it binds it at the active site, but fibrin is now inactivating the thrombin as a negative feedback loop. And eventually the clot to get to a large enough size that you're not generating thrombin anymore. And that ends the coagulation, limits the clot size. The other thing I'll point out here is, notice again, you need calcium, and there's a couple clotting factors. Now, one of those clotting factors I actually name here, and the book does as well, without using a Roman numeral. I give it an actual name, and it's called fibrin stimulating factor. This was released by not the liver in this case, it's made by platelets and released by the platelets. So once again, platelet activation and degranulation has to happen for you to get a successful blood clot. So the role of this is to take these long fibers and to interconnect them so that you end up with a mesh, and they're bound to each other. They're not going to just flow out with the blood that tries to pour out through the opening if the platelet plug becomes unstable. You need to make them into this elaborate mesh, and that's important for having a long-term clot. So here they're actually giving you the Roman numerals for it. You need calcium in order to make this fibrin stabilizing factor do its job, but it makes the fibrin become this cross-linked fibrous mesh. Okay, so lots of positive and negative feedback loops. So how do we limit clots to being a certain size, or prevent their inappropriate clot formation? Well, the last group is going to talk about how you prevent these inappropriate or also too big of a blood clot. You may not have talked about that, but I can. So who is the spokesperson for group G?
That's me.
Okay, Dina?
So question 18 is, what roles do endothelial cells lining the vessels play in preventing inappropriate clot formation and in limiting the size of the clots that do form? Nitric oxide and prostacyclin are released by healthy endothelial cells to prevent platelet activation and prevent the formation of inappropriate clots. The fibrin stabilizes the formation of clot sizes by absorbing and or binding to the free thrombin, and the removal or inhibition of clotting factors like antithrombin 2, heparin, and von Willebrand factors slow and or stop coagulation.
Great, that's perfect. So remember that prostacyclin and nitric oxide are also vasodilators. So that means that if you if you have a healthy blood vessel, healthy capillary or healthy arterial or venule, you are constantly releasing some prostaglandins from these healthy endothelial cells, but you also may release, if you want blood flow into that area, vasodilators locally. And those vasodilators will keep blood flowing, but they'll also help suppress the formation of clots. So once again, we run into this umbrella term of prostaglandins that go unnamed, and one of them is prostacyclin, but there are apparently other things released by healthy endothelia to say, cool it, guys, you don't need to clot, and suppress platelets from encountering each other by keeping the blood flowing in a continuous way so the traffic doesn't get clogged up. So von Willebrand factor apparently plays partly that role as well, as long as there is no collagen around to bridge. And if you have an undamaged vessel, then you have no collagen around, and then it helps to suppress them. Now, the one other thing, and I think you mentioned this, maybe not, I can't remember if I thought about it or you mentioned it. The plasma has one of it's probably one of its alpha or beta globulins, again, is called antithrombin.
Yes, I mentioned that.
You did mention it. Yeah, so that's already in the plasma. And so you, again, have this sort of terrible balance between trying to clot and trying to activate platelets, and no, don't clot, don't activate platelets. And what tips the balance is things like the slowing of the blood flow so that you build up platelets near each other and activate some, or collagen exposure, or hydrolytic enzymes that are released by either the platelets or damaged tissue that can start to release signals to cause clot formation. So before we leave this, I just wanted to point out again in this figure that if you read the textbook, they don't use PF3. This stuff right here is the same as PF3. And, you know, then they kind of do both. To me, this new edition does the worst of both worlds. In the figure, they become really vague and say phospholipid on the surface. Well, that's PF3. And then in the text, they mention this phosphatidylserine, which would be hard to say. So that's where I prefer. But, you know, if you use these other terms, this is the more dangerous one because there are many phospholipids on the surface of aggregated platelets, not just this one. It has to be this specific one. And then they don't show its role down here in this figure, which it does have a role again, the PF3 down there in that figure. And then once again, I'm just pointing out, you don't need to know the Roman numeral 13 here. You can just call it fibrin stabilizing factor, but I would know that you can't make a long-term clot without this, and that this is from platelets, not from the liver. So one other thing, just as an interest, I think I mentioned this during the lecture, vitamin K is critical for the production of these clotting factors by the liver. So vitamins in general are usually coenzymes that help a cell produce what it needs to do its metabolic pathways. So if you don't have enough vitamin K in your diet, then your liver is unable to make this myriad of Roman numerals that you're seeing up here, and your blood is poorly clotting. So that's just more of for interest. You may have heard of the blood thinner. They call it blood thinners. Blood thinners aren't really making your blood less viscous. What they're doing is making it less likely to clot. I'm not sure how they got the name thinning on it, but Coumadin is a common one that people with poor blood flow might have, or heart patients where they may need to, or say people that have major surgeries may be given various forms of blood thinners. But these are acting by interfering with, Coumadin itself acts by interfering with vitamin K. So that means that you're not just targeting clotting at the area of the surgery, you're targeting all blood clotting. And people taking Coumadin do have a danger of having poor clotting and losing too much blood to a major incident where they're hemorrhaging.
Professor? Did you want number 19 answered as well?
Yes, so we're going there right now.
Oh, okay. I thought you forgot about it.
No, I haven't forgotten it.
You were wrapping up the lecture. I was like getting confused. It's okay.
All right, I just figured I have this
You know when you're ready.
Use it. So now that we've got this big old clot here, we already answered the question here, but what for 19 helps to eventually break this clot down?
There's a couple of things. Plasminogen is made by the liver, and it's a protein, and it is actually incorporated in the fibrin mesh of the clot. The healthy or healed endothelial cells release TPA, which activates the plasminogen, and then plasminogen digests the fibrin over the next several days as TPA slowly reaches the plasminogen embedded in the clot.
Great. The only thing I'll correct is that when you see the word ogen, that means the inactive form. So what you want to do when you activate it is get rid of that part. Okay. So the TPA helps convert plasminogen into plasmin.
Okay, yeah, I have plasmin in my notes and thought I mistyped, so.
Okay, great. So you're using plasmin to digest the fibrin. So the clot itself already binds up this plasminogen that's in the plasma. The liver made it. It's, again, one of those alpha and beta globulins. And as the endothelia cells heal, they make that TPA, and the TPA starts to activate the plasminogen. So good. All right. And TPA is, as the slides tell you, can be used in patients with strokes to help them get over the stroke. It prevents too large a clot from forming, and therefore, the person would have greater blood flow, say, to their brain. But remember, it's acting at clots everywhere. So if that person is at risk for thrombosis or potentially has a thrombosis somewhere in their body, and you start giving them TPA because they could potentially, be having a stroke. So you really can't willy-nilly give TPA. You have to know more of the patient's history before you go giving TPA. Okay, so we finished these lecture questions. Let me stop sharing this. And I'm going to go back to sharing my iPad because what I did was summarize. I got to wake it up first. And I know what slows it down. When I get a text, it's like that takes over my life and it takes over my iPad, and that's why it goes really slow. So I'm sorry. Let's see. Okay. So I kind of listed some of the things that we encountered as roles of the endothelial cell. Going back, this is part of our review in a way. Going back, the endothelia are listening to these conditions of the tissue. Does the tissue around it have a good level of oxygenation, or is there high CO2? The opposite, right? Low CO2, high oxygen, low oxygen, high CO2. Is there a lot of potassium around in the case where you're low oxygen as well? Is there a lot of H+ ions around? Is it low pH? Those would all be poor oxygenation. And these endothelia are listening to these conditions and then releasing local vasodilators. So I'm going to move you guys over here so I could see the whole screen. So in response to these conditions, they release things like nitric oxide and prostacycline and adenosine, or ATP even. These are released and they cause local vasodilation. Again, you don't need to know the specific prostaglandin, but they can also release prostaglandins, and prostacycline is an example of a prostaglandin, but there's another one, prostaglandin 12, and it is, again, all vasodilating. Now, what I did with these arrows is I pointed out that some of these things also prevent platelets from aggregating at the same time. So they're sort of a twofer here, they can do those. Act on the second thing. The second thing we'll look at that endothelia play a role in, which is blood clotting. If conditions are low CO2, high oxygen, low potassium, and more normal pH, then local vasoconstrictors can be released. And one of those is called endothelians, a group of them actually, endothelians. And I keep leaving the A off of this. This should have an A on it. Thromboxane A is also released. Again, it plays a double role because it helps platelet aggregation. Think about the situation here. If you have damage and you're bleeding out in that area, then your tissues are going to get low oxygen. I'm sorry, that's the opposite. I'm sorry, forget that. I'm talking off the top of my head here. Anyway, thromboxane A does play a role in both the aggregation and encouraging vascular spasm. So you see that with the vascular spasm here, it's a vasoconstrictor. Okay, then we go on to the second roles that endothelia play, and they clearly aid in preventing platelet aggregation because they're physically covering and hiding the collagen. And then I also will say, and they release, von Willebrand factor and other signals to prevent platelets from aggregating. So they're sitting there as healthy endothelia. They're releasing these signals to prevent any aggregation from occurring. If you do get disrupted endothelial lining, then they're going to aid platelet aggregation. They're going to increase whatever granules they have of von Willebrand factor, and that's going to act as a bridge with the collagen, because without the collagen being there, von Willebrand factor can't help the aggregation. When you have no collagen exposed, von Willebrand factor actually limits aggregation, which kind of is sort of amazing when you think about it. Eventually, you'll get a clot formed, and when the clot is formed, the healthy endothelia will now release TPA, and that will that's what it stands for, TPA, tissue plasminogen activator. So it makes plasminogen go into plasmin, and plasmin digests the fibrin. Now, I put a dashed line here because you don't need to know this yet, but what we're going to see is that they'll also play a big role in our immune response and enabling both inflammation, which means what I mean by both increased permeability and vasodilation that occurs in inflammation. And they also release something called heparin, and heparin is an anticoagulant that you will want initially when there is a infection, because what you're going to do is use the bleeding out of that damaged area to try and flush. So you're going to try and force a current to keep the pathogens from getting into your bloodstream. So when you actually have a infection, when there's a pathogen there, you'll see that they play a role in preventing it from getting worse. Okay, what time is it? 11:03. Oh my God, I'm sorry, I went way over here. I guess all our review will be in lab, which is just in 7 minutes. I thought we would finish before this, but I go on and on, don't I? So let's see what I have in my chats. And most of it's about sharing my drawing. Oh, thank you, Dina. She's saying I can put my iPad on Don't Disturb airplane mode. I think I can do that. I mean, I know I can do that. I want to make sure I can still use my wireless connection to my computer. Okay, guys, so you did a great job with all of this. Hopefully you've got lots written down. And if you'd like, I will share this summary since it's a nice little bit. I'll put that on, like I did before, I'll send my summary. I'll stop sharing here so you don't have to see all that. And I will see you in just maybe 5 minutes, and we can do, what we're going to be doing in lab is going over the blood pressure handout that's due today, as well as a review for the lecture exam. There's no new lab material. Okay, I will see you shortly and take a little break.